a super fast version of the D1 unit, starting with D1.1 DNA replication. Let's jump in. So why do we need to do DNA replication? Number one, we need to replicate DNA before it divides through meiosis in order to produce sex cells and be able for reproduction to occur. Secondly, we are constantly needing to grow and replace tissues. In order to do that, we need to replicate DNA before cells can divide through mitosis. So the basics of how it occurs. Here's our original strand. We're going to call these each parent strands. Okay, as the replication fork forms during DNA replication, we're going to see that each of these resulting identical copies contains one of this parent or template strand and one new strand that's been formed nucleotide by nucleotide in order to have now two identical pairs. Okay, DNA is replicated semi-conservatively, which means that each of the resulting DNA strands is made up of one parent and one new strand. These two forms were both not found to be biologically significant. Okay, a reminder of our base pairing rules. Adenine always pairs with thymine. Cytosine always pairs with guanine. Now, you don't need to memorize the number of hydrogen bonds that are formed between these base pairs, but you need to know that the reason they will only bond with each other is because they want to make a certain number of hydrogen bonds. All right, now for SL and HL combined, you will have to have two enzymes that you need to know. And then for HL only an additional, okay, you're going to have several more. So let's begin with our two important enzymes. Our first one is helicase. Helicase kind of looks like a donut. It's round and there's a hole in the middle, and its job is to move along the DNA strand and separate it. It's like a zipper that's going to unzip it, and it's going to break the weakest bonds, which are those hydrogen bonds holding together the complementary base pairs. Remember that we have a sugar phosphate backbone made of covalent bonds, so that's a lot stronger, and that's going to stay in place during this process. Then we have our second important enzyme. Now remember that a polymer is made up of many monomers, and ACE refers to an enzyme. So anytime we're talking about DNA replication, you're going to see that DNA in front of it. And then here we have polymerase. It's an enzyme that makes a polymer. So DNA polymerase's job is to take these monomers, these nucleotides, and polymerize them to form covalent bonds that allow them to be in a long row of called a polymer instead of individual monomers. So its job is to add nucleotides one by one along the parent strand using those complementary base pairing rules. All right, polymerase chain reaction is a method that scientists can use to amplify DNA. That means we're going to make many copies of it. Okay, and this is a thermal cycler, which we're going to use to do so. It's going to switch between 3 different temperatures, and at each of these specific temperatures, okay, that we stay at for a specific amount of time, we're going to be able to move forward in our DNA replication. And since we can do many cycles, okay, and we have exponential growth, so say we started with a single strand of DNA, after one cycle we would have 2 strands, okay, but then after another cycle we'd have 4, and then 8, and then 16, and so. Okay, and 30 cycles can amplify the DNA by a factor of 1 billion in under an hour. All right, so in addition to that sample of DNA that we want to amplify, we're also going to have a lot of free nucleotides as well as DNA primers. Okay, these primers are going to be specific sequences that bind to our target DNA sequence, okay, on either end of the sequence that we are trying to replicate. Usually, DNA amplification is somewhere around 100 base pairs, a piece of the DNA that we're going to want to be able to have millions of copies of, okay, in order to do whatever we're going to do with it. And we'll cover some important reasons we want to do this in a bit. Now, we're going to be doing this at very specific heats, and so we're going to need a special enzyme. We know that as enzymes get too hot, they begin to denature, and DNA polymerase from humans at the temperatures we're going to be doing thermal cycling at would denature. So, what scientists have done is they're using the DNA polymerase from a bacteria that is adapted to live in the very hot springs at Yellowstone. And that TAC is just short for Thermus aquaticus, the name of that bacteria. Now, because it can live in such a hot place, okay, those enzymes in the temperatures that we are using in a thermocycler are not going to denature. So let's run through the 3 steps. The first step is we're going to heat it up to 95 degrees Celsius for a short 30 to 60 seconds. Now, I wouldn't memorize these specific numbers, just I would outline the general process. So we're going to heat it up. This first step we're going to call melting. Now, if we're heating it, it's because we're trying to break bonds. And the first bonds are going to be broken are the weakest ones, which are going to be those hydrogen bonds. So we're going to see that this melting step breaks the 2 strands apart at the hydrogen bonds, but this specific temperature and time, okay, is not going to break the stronger covalent bonds that form those sugar phosphate backbones. So we have this very specific temperature that allows for just the weaker bonds, the hydrogen bonds, to break. Next step, annealing. This is where we're going to cool it way down to 54 degrees Celsius, which is what's going to allow for those DNA primers to bind that are going to be the starting points for our third step, which is elongation. And this is where that TAC DNA polymerase, okay, is going to move along that parent strand, adding free nucleotides that are in the thermocycler that have been added, okay, and we're going to end up with a copy of every single strand of DNA that was in there. Okay, now at that 95 degrees Celsius, that hot point, any like regular human DNA polymerase would be denatured. And that's why TAC polymerase is special, is that it can, even though its job is here in elongation, right, it's still present throughout the whole cycle. So it won't denature at this melting point. Here's just one more image to kind of give you another way of looking at these 3 primary steps. Okay, gel electrophoresis. This is a technique that can be used to separate fragments of DNA by length. Okay, this gel provides a medium that the DNA is going to move through. And when it's moving through a gel, we're going to see that smaller fragments can go faster. Larger fragments are going to take more time to migrate through. I like to think of people trying to run through like a dense bamboo forest, right? If you have someone small, right, like a kindergartner, they're going to be able to scoot in and around all of the branches much more efficiently and run really far compared to like a sumo wrestler who's going to have to really, take their time to try to find a way through all of the branches. So large sumo wrestlers are not going to be able to travel as far through the gel medium. Okay, and we're going to see the reason it travels at all, okay, is because DNA is negatively charged. And so if we can set up a negative and a positive electrode, okay, we're going to place this gel in an electrolyte solution and we're going to turn on our power supply to apply a voltage. And we're going to see that our negatively charged DNA is going to be placed at the negative end of the electrode so that it will migrate across the gel towards the positive electrode because opposite charges attract. And sure enough, we'll see those shorter fragments are able to migrate more quickly than the larger ones. Okay, oftentimes we'll also see what's called a ladder in gel electrophoresis. Okay, this is just going to be a sample with fragments of known lengths, and it can be used for comparison to estimate the number of bases in each of these other fragments based on how they align with this ladder. Okay, after the gel is run, it gets treated with a dye to make DNA visible. Okay, and then we can compare different samples to each other and look for matches. Okay, so a couple of applications of PCR. Testing for coronavirus. Okay, now this is going to be a test that can look at very, very small samples. Very early in the infection process, it will be detectable because the PCR can amplify even a very small sample of viral DNA millions of times, and so we can pick up infections that only are just getting started. Okay, disadvantages of testing this way instead of, say, an instant test. okay, is that generally it needs a testing laboratory and it's going to be more expensive. And results may not be immediately available because it has to run through that thermal cycle. Okay, another application is going to be DNA profiling for paternity testing. Okay, and this is how, if it's unknown who the father is, paternity testing can help figure that out. So let's look at a couple of practice problems of when this can be useful. So first up, in forensics. Okay, say you were given next to this ladder that A is going to be the well where we had some DNA trace evidence from a crime scene. B is a homeowner, so we can make sure that it's not just the homeowner's DNA that's being picked up. C and D are both suspects. And so we can look for where the matches are to the trace evidence. And you can see here that suspect 1, which is in the C column, has the exact match as the trace evidence. Okay, so we can knock the homeowner out and suspect 2 out as suspects because their DNA fragments are not aligning with the trace evidence. Okay, so what if this was a paternity question? Here we have a mother, okay, and here we have the child, and then there's 2 different men that are being tested for paternity. What we're going to see is that the child is going to have fragment sizes of either mom or dad. So here's the child in column B. Here we're going to see that this fragment size is the same as column C. Okay, and then this one, it doesn't have C or D, but this fragment size is the same as mom. Again, this one is the same as C, this one is the same as mom. And so we can determine that it is most likely man 1 who is the father of this child. Okay, at this time, if you are an SL student, you're going to pause and skip ahead to D1.2. HL students, we still have just a bit more. Okay, a quick review. Remember that on a pentose sugar, we number these carbons clockwise starting from the oxygen. Carbon 1, 2, 3, 4, 5. Okay, and then we use those carbon numbering to be able to talk about the directionality of RNA and DNA. So we call this the 5' end where carbon 5 is, and this is the 3' end. I had a student suggest to me yesterday a great way to remember this, okay, is that this kind of looks like this sugar is like giving a high five. Like if this was the arm, like with this little hand giving a high five, just to help you remember that the phosphate end with the little high five here is a 5' end. So here's our phosphate, there's our 5' end. I thought that was great. All right. Now, here's our replication fork. We're going to see that DNA replication always occurs in a 5' to 3' direction. And you're going to want to remember that because that is going to be the same direction for both DNA replication and transcription and translation. Everything always goes 5' to 3', and they love to ask that. So just remember, it's always 5' to 3'. Okay, and that's going to be in DNA replication, the directionality of the strand that's forming. So here on the leading strand, that's going to be no problem at all because we're going to be adding nucleotides one by one as we move towards the replication fork as the fork opens. But on our lagging strand, we have a problem. We can still only add from 5' to 3', but we're going to have to add in fragments, and those are called Okazaki fragments because 5' to 3' leads away from the replication fork. Okay, you might see a question where you're given a diagram and you're asked, where would the next free nucleotide be? Okay, so you're going to want to look for a free 3' end where you can have another little high five added, and that would be your 5' to 3'. So it would be right there. Okay, I think we've pretty much covered all of this. So let's look now at those additional enzymes. So we already talked about helicase with our SL counterparts. Okay, but let's look now at a few more things that have to happen, a little more depth. So DNA primase. Remember, if there's DNA, we know it's going to be used in DNA replication. And just like it sounds, ACE, it's an enzyme and it lays primers. Now, these are actually RNA primers. They're indicated here in red. So DNA primase is going to be placing DNA primer, or excuse me, RNA primers, it's only going to need to place it once on the leading strand, okay, because this is going to be where DNA polymerase 3, our next enzyme, is going to start adding nucleotides. Okay, we're going to need several RNA primers placed, one for each of our Okazaki fragments on our lagging strand going the opposite direction. So now that we have our RNA primers placed by DNA primase, this is where DNA polymerase 3 comes in. Now this is the same one that we just called DNA polymerase earlier when we were with the SL students. Now DNA polymerase 3 is super important because it does two things. Not only does it do that important job of polymerizing, adding individual monomers, okay, making it into a polymer one by one, adding nucleotides in each direction, adding these green colored ones, but it also has a secondary job of it proofreads each nucleotide after it has been added to the chain, okay, and if it's the wrong one, it can switch it out. After DNA polymerase 3 has added at what you can see here in green, okay, we have a little problem because we've still got some RNA mixed in here indicated in red. And so that's where our next enzyme, DNA polymerase 1, comes along. Annoyingly, the numbers aren't quite in order. Don't worry about it. You just need to know that polymerase 1 is the one that is then going to remove the RNA primer. It's an exonuclease. Okay, but then it's also going to replace those RNA nucleotides with DNA nucleotides. Okay, so it's going to remove all of these reds, turn them into greens. Okay, but there's going to be a gap that's left because DNA polymerase can't make a 3' to 5' link when it reaches the next Okazaki fragment. So there's going to be a gap. So that brings us to our final enzyme, which is DNA ligase. I like to think of ligase like a glue stick because its job is to go in and seal all of those gaps that are left. Okay, ligation means to join things together. Okay, so here's just an overall, if you want to pause and run through each of the roles of each of those different enzymes. Okay, so again, just remember that DNA polymerase 3 is going to recognize base mismatches. It excises the incorrect nucleotide and inserts one that has the correct base, and we call that process proofreading. Okay, on to D1.2, protein synthesis. Okay, we need to keep straight everything we just covered is DNA replication. Now we're going to talk about how DNA is used to make proteins. This is our central dogma of molecular biology. DNA is our master plan. We have one copy of it and it's safe in the nucleus. Through the process of transcription, which occurs in the nucleus, DNA is made into copies called mRNA or messenger RNA. That messenger RNA then exits the nucleus through a nuclear port, okay, into the cytoplasm. And there it goes through translation, where it builds a polypeptide of amino acids and that folds into a protein. So DNA makes mRNA, makes protein. Okay, this is why you have DNA, is to code for protein. So this is a very important process. Okay, so let's start with talking about transcription. This first step, taking that master plan and making copies of it. Okay, DNA is double-stranded. It makes single-stranded mRNA, and we can make as many mRNAs as we want to from that DNA template. Okay, now we have another enzyme. This enzyme in transcription, though, is making RNA, so it's called RNA polymerase. All the ones from the last subtopic were DNA. This one is RNA, and it's only one, and it does pretty much all the jobs that it could possibly need to do. Okay, it starts by binding to a site at the start of the gene that's being transcribed on the DNA. Okay, it does the job of helicase, where it unwinds the double helix. It moves along the template strand. Okay, and then it does the job of DNA polymerase 3, where it positions RNA nucleotides on the template strand with bases complementary to those of the template. The difference, of course, being that it's RNA nucleotides. It links them all together to form a continuous strand, and then it detaches the RNA from the template strand and allows for the DNA double helix to reform. Now, whereas DNA replication, you're replicating the entire strand of DNA, transcription is just going to replicate a portion of the DNA that codes for a particular gene or for a particular protein or proteins. All right. So we call this strand that's going to be used as a template the template strand. But then we also have the sense strand, which isn't the strand used as a template, but it's going to look exactly like the RNA that's going to be made. The only difference is the RNA is going to have uracil, where the sense strand has thymine. All right, just note that DNA is very, very stable. Its particular molecular structure allows it to just be extremely stable, and that's why I think life has persisted as it has. Okay, this term is an important one, gene expression. Gene expression is which proteins get made from your DNA, because every cell in your body, with a couple of exceptions, has your entire genome. All the DNA is in every single cell. But not every cell needs to make every protein, right? If you're a neuron, there's no reason for you to have hemoglobin to be able to carry oxygen. So you don't want to be able, or you don't want to make every protein in your entire genome. Like, that's just a waste. It's inefficient. And so we have gene expression, okay, where only certain select genes are produced depending on the needs of the cell and the job of that cell, okay, and the time in that cell's life. So transcription is where we can switch on and off which genes are being made into proteins at any given time. Gene expression. Okay, so let's move on then to translation. So once that mRNA has been made, it exits the nucleus into the cytoplasm. Okay, here it's going to meet with a ribosome. Ribosomes do protein synthesis, so this is where we're at. Okay, let's look a little bit more closely at how the ribosome is going to turn this code, this mRNA, into a chain, a sequence of amino acids. So it's going to need some help. Okay, first some vocab you should know. Every 3 bases on the mRNA are called a codon, and these are going to code for 1 amino acid. Okay, so this goes for an amino acid, an amino acid, amino acid, every 3. Now the codon is going to be complementary to an anticodon on a tRNA, which stands for a transfer RNA. Now a tRNA is going to have a specific amino acid bound to it. And so all of this is very specific. This anticodon is only going to bind to the complementary codon. Okay, and we're going to see that it transfers or drops off a specific amino acid that is going to be coded for, therefore, by the codon. So we're going to see that mRNA has a site that we call the start codon, and this is going to be the codon AUG, and it's always going to code for the amino acid methionine. Okay, and then we're going to have every series of 3 bases called codons, which each code for an amino acid, until we reach one of 3 stop codons that is going to indicate that that's the end of the sequence. Okay, tRNA, okay, in reality looks kind of like this. You should recognize it. Okay, I kind of think of it looking like a, I don't know, maybe a big, like a drill, or like a little alien, like space invaders gun, like pew, pew, pew. Okay, but a 2D model is going to look like this, and we're going to see that it's made up of these 3 loops, one of which has the 3 bases that make up the anticodon. The note on the other end is going to be the amino acid attachment site. Okay, ribosomes are made up of 2 subunits, a large and a small, and I think of them like hamburger buns. The large subunit is going to be where the tRNAs are going to bind. Okay, the small subunit is below, and the mRNA is sandwiched between the 2 buns. We're going to see that tRNAs are going to bring their anticodon to match up with the codon and be able to drop off the amino acids. And so everything is based on complementary base pairing. Okay, and we can determine which amino acid is going to be brought, okay, using an amino acid chart like this. For example, if you were told the codon is GCG, you would look for the first base here, G, you would look for the second base here, C, and then the third base here, G, and you would say that that was alanine. Now, interestingly, you can have multiple different codons code for the same amino acid, and we're going to see a little bit later why that's important. Okay, let's see. I think we've coded all of these. Just to note, here's our start codon. Here's our 3 stop codons. Everything else is going to code for one of our 20 amino acids. Okay, now this property I just mentioned, okay, we refer to the code as degenerate because different codons can code for the same amino acid. Okay, and then there are some practice questions here that I'm just going to flip through because this should not be the first time you're going through this. This should be your fast-paced review. So hopefully you can answer all these questions. Note that you should also be able to use one of these round amino acid tables, and in this case, you always just go from the inside out. Okay, so I think I said GCG, if I remember correctly. So you would start at the inside and find the G, then the C, then the G, and sure enough, there's alanine again. All right, so let's talk about the process of translation. Now, what's going to happen is this ribosome sandwich is going to move along the mRNA in a 5' to 3' direction. There it is again. Okay, and as it moves, it's going to open up this A site for another tRNA to bind that is complementary to the codon. Okay, and then as it shifts another 3 bases, okay, that tRNA shifts over to the P site and then eventually to the E site with the next shift. Okay, and as we see this happening, we're going to see that each of these amino acids, okay, is being dropped off. The growing polypeptide is added to the end of it, okay, and then that tRNA that gets released in the exit site is going to be able to go get recharged up again. Okay, what does it mean to be charged or activated? That means that it's going to have its specific amino acid added to it. Okay, and you should know that there are specific activating enzymes for each specific amino acid to be added to its specific tRNA that it can bind to. Everything's very specific, but once this gets added using this enzyme, we now say that it is charged or activated. Okay, so let's run through the process. A tRNA carrying a single attached amino acid comes in and binds to the A site. This is our first site, just like A is the first letter of the alphabet. tRNA binds, its anticodon is complementary to the codon on the mRNA. Next, the single amino acid on the tRNA is linked to the end of the growing polypeptide. Okay, by the formation of a peptide bond, the tRNA is now holding the whole of the growing polypeptide. The tRNA moves from the A to the P site as the ribosome shifts in this 5' to 3' direction. The anticodon of the tRNA is still paired with the codon on the mRNA. Now, if you are asked to draw an arrow to show the ribosome's direction and you're shown the 5' and the 3' end, okay, sometimes IB likes to be tricky and they'll put a 5' here and a 3' here. So be ready to show an arrow pointing to the left. If it's like this, 5 to 3, then show an arrow to the right. But you need to not assume that it's just going to be read like a book, okay? But you need to look for the 5 and the 3 and know that it's going from 5' to 3'. Okay, picking back up again. Okay, the polypeptide held by the tRNA is transferred to another tRNA that arrives at the A site, and we keep building this polypeptide. Okay, and every time it shifts then, that tRNA that moves to the exit site that's dropped off its amino acid is then able to go find another enzyme to get activated again, and the cycle continues until a stop codon is reached. Now, let's talk about gene mutations. A mutation is just a change to the base sequence of a gene, and it can be as small as a single base substitution. Okay, here you can see that just a single base changed, and in this case, it coded for one wrong amino acid. Now, this could have very little effect, or it could have a massive effect. Okay, it just depends on that particular polypeptide. Now, an example of where a single base substitution has a massive effect is in sickle cell disease. Here you can see that where the DNA would normally have read G or CTC, it reads CAC. This single base change means that valine is produced instead of glutamic acid. Now, one amino acid here is enough to change the structure of hemoglobin. Let's see why that is. Glutamic acid, note, has a negative charge on its R group. Valine is nonpolar, there's no charge. That is enough to change the physical structure so that when we have normal hemoglobin, it's this shape we've seen before with these 4 polypeptides and 4 heme groups. Look at this sickle hemoglobin, sorry. Look at how different this shape is. It's so different. And this is what then causes those sickle-shaped cells, okay, which can lead to a lot of issues with getting kind of caught as they're trying to travel through narrow capillaries, okay. Now, interestingly, we still see prevalence of this sickle cell allele because if you only have one copy, if you're heterozygous, okay, then you only have this mild anemia, but it also provides that malaria resistance. And so it's persisted in the population because of the heterozygote advantage. All right, that takes us to the end of SL content for D1.2. HL students, let's keep charging ahead. All right, remember, directionality of transcription and translation, just like DNA replication, is always 5' to 3' direction. Okay, a promoter is a section of the DNA upstream of the gene that initiates gene transcription. Okay, it could be between 100 and 1,000 bases long, and it's located adjacent to those genes. Okay, the base sequence is going to allow for that RNA polymerase, that enzyme that does it all, to bind along with a variety of proteins that act as transcription factors. So we could either have repressor sequences within the promoter that allows those factors to bind, okay, and that when they bind, they prevent binding of RNA because they're repressing it. Or we can have activator sequences that do the opposite. When transcription factors bind there, they encourage the binding of RNA polymerase, leading to transcription of that gene. Okay, now depending on the stage in that cell cycle, okay, what its particular job is at the moment, they may change their pattern of gene expression. Some genes also have similar promoters, which means that they all get expressed together, even if they're on different chromosomes. Okay, here are some examples of non-coding sequences in DNA that don't code for polypeptides. Okay, but they do have other functions. So for example, we have some base sequences that are transcribed to produce those tRNAs, those transfer RNAs. Base sequences that can be transcribed to produce ribosomal RNA. This is what actually makes up the ribosome. Ribosomes are made of rRNA and protein. Some base sequences can be used in the regulation of gene expression, like the promoters we just mentioned. Okay, telomeres are structures that form on the ends of chromosomes in eukaryotes, and they act as like a protective cap. And every time cell division occurs, those telomeres get a little shorter and a little shorter. Okay, but they do not contain DNA that actually codes for any proteins. Okay, and then introns. We're going to talk about exons and introns here in a minute. We're going to see that processing occurs of that mRNA before it leaves the nucleus into introns and exons. And while those exons are going to actually exit the nucleus and be coded into proteins, introns go into the recycle bin, as we say, where they can get broken down and their nucleotides reused again. Okay, now that process I just mentioned can't happen in prokaryotes because there's no separation, there's no nucleus to separate that mRNA from the ribosomes. And so there's no opportunity for that modification. And so here you can see that transcription and translation are occurring at the same time. Okay, so let's look at some examples of that modification that can occur in eukaryotic cells. So when RNA gets modified, okay, a 5' cap is going to be added to the 5' end, okay, and a 3' poly A tail which is between 100 and 200 adenine nucleotides, are added to the 3' end. Translation stops before the ribosome reaches the poly A tail, and the function of the 5' cap and the poly A tail is to stabilize the ends of the mRNA by protecting them from digestion by nuclease enzymes. They also encourage further modification of the original transcript. Okay, so here we go. Exons and introns. Exons are going to be expressed. They exit the nucleus and go on to meet up with the ribosome and make proteins. Introns are not expressed. They go in the recycle bin. Now, your genome, one particular gene can make many proteins. So we can see through alternative splicing. Here we have 5 different exons and we have some intron regions in between them. But sometimes an exon can get excised along with those introns. So for example, here, exon 3, right, has been removed along with those adjacent introns. And so we're going to get a different mature mRNA. Okay, and that's also done different from this one where exon 4 was removed. So that means that this one DNA sequence can code for multiple different proteins depending on how that alternative splicing occurs. All right, so let's look at translation again, okay, but now with a little bit more detail. So let's first talk about how translation is initiated. Okay, so first up, we're going to see that an activating enzyme attaches an amino acid, the amino acid methionine, to the initiator tRNA with the anticodon UAC. That's a repeat. We already talked about how every tRNA has a specific activating enzyme, okay, that's going to activate a specific amino acid. Okay, but here we go. So now this is new. Our initiator tRNA, so that's the one that has that methionine, okay, and the anticodon that is complementary to AUG. Okay, it's going to bind to the small subunit of the ribosome. So we start with the bottom bun, just like we're building a burger. We're going to start with the bottom bun. The small ribosomal subunit then, with that initiator tRNA attached, is going to bind to the 5' end of the mRNA. And it's going to start sliding along it, and it's going to use that UAC anticodon, which is complementary to our AUG, okay, and it's going to move along until it finds AUG. Next, okay, we're going to see the small subunit's going to stop moving because we now have complementary base pairing that can occur from that UAC anticodon, okay, to this AUG codon. This, now that it's been found, is going to allow for the large subunit to come and bind, okay, with that initiator tRNA binding, falling right into that P site, that middle peptide site. Okay, so we're going to now see that a peptide bond is formed. I think I skipped a step. I'm sorry, I'm going to go back. Sure enough. A tRNA with an anticodon complementary to the next one moves in. So this is where we pick up with where we were with the SL students. Okay, and so just to review this again, we form a peptide bond between these 2 amino acids. Okay, it's held by the amino acid in the A site, and then the ribosome shifts over 3 nucleotides. Okay, eventually another shift until each of our tRNAs leave through our exit site, and we have our growing polypeptide, and that will continue until we reach a stop codon. Now, we can also, in addition to having modification of the mRNA, we can have modification of the polypeptides after translation occurs. So, some examples of modifications include removal of the amino acid methionine from the 5' end of the polypeptide, changes to the side chains of amino acids. Okay, the polypeptide can be folded and we can make that tertiary structure. Okay, for example, disulfide bonds can be formed between cysteines. Okay, we can convert propeptides into mature peptides by removal of part of the polypeptide chain. We can combine 2 or more polypeptides into a quaternary structure, like with our old friend hemoglobin here. Okay, and we can also combine non-polypeptide components into a quaternary structure of conjugated proteins, again, like hemoglobin that has these non-polypeptide heme groups. So let's look at an example that IB would like you to be familiar with. because it actually contains a lot of the list on that previous slide. So we're going to look in more detail at the modification of pre-proinsulin to insulin. So first up, we know that insulin, is produced by pancreatic beta cells. And those pancreatic beta cells are making insulin for excretion. And so they're going to be using ribosomes that are on a RER, because that allows for that modification that we're about to see to happen inside the lumen of the RER, the inside. And we know that vesicles then can bud off containing insulin, get processed by the Golgi, and then released out of the cell to be transported through the bloodstream to every cell in the body to let them know it's time to take some glucose out of the blood. Okay, so let's break down then, how is insulin modified into its final state that allows it to act as that messenger hormone? Okay, so first of all, when mRNA is translated by ribosomes, the polypeptide produced is called pre-proinsulin, and it has 110 amino acids. That pre-proinsulin goes into the lumen of the RER, and here we're going to see a sequence of 24 amino acids removed by a protease enzyme, which cuts proteins. A chain of 86 amino acids remains, and it's now called proinsulin. So we've just cut off 24 of the original 110. And that takes it from pre-proinsulin to just proinsulin. Next, we're going to see that proinsulin becomes folded with 3 disulfide bonds stabilizing the tertiary structure, indicated here between cysteines. Peptide bonds are broken at 2 positions in proinsulin by proteases that recognize pairs of positively charged amino acids. This causes a sequence of 33 amino acids to be excised, and it's going to leave us with 2 separate chains: the A chain with 21 amino acids and the B chain initially with 32 amino acids. These 2 chains are held together by those same disulfide bonds that we saw earlier. Lastly, a pair of amino acids is going to be removed from the C terminal of the B chain by a protease to yield mature insulin with a total of now 51 amino acids. So we've gone from 110, to less than half of that, 51. We've processed it, so insulin is now 2 polypeptides, bonded with these 3 disulfide bridges, and it can now be sent to the cells in the body, as a chemical signal. So here's just another image of it. Here's those 3 disulfide bonds, and you can see all of these different chains. I don't think you're going to need to memorize what the names of all the chains are or anything, but just be able to recognize the process of post-translational modification in order to turn a polypeptide into its functional state, like insulin. And then lastly, to wrap up this particular slideshow, we're going to see that when those proteins are either damaged or just getting older, that we're going to want to recycle them. And we just recycle them using proteasomes. We can break them back down into shorter chains of amino acids, which then can be fully digested into amino acids and can be reused again to make new proteins. Okay, we've got one more slideshow to go. D1.3, mutation and gene editing. Let's jump in. So we already mentioned that a gene mutation is a random change to the base sequence of a gene. So let's look at some different types of gene mutations. First up, substitution. This is where one base is replaced by a different base. This can be caused by chemical changes to bases or by mispairing during DNA replication. Now, this may or may not cause a new amino acid to replace the old amino acid. Because we know that this is a degenerate system, sometimes you can have a whole mutation where you have a new base, but it will still code for the exact same amino acid. And we're going to look at that in a second. Now, what happens though if instead of just a replacement, instead a nucleotide gets inserted, so there's an extra base? This is a bigger problem because it means that everything is going to shift over. Whereas instead we might have just had one amino acid be different. Now Nearly every amino acid is going to be different after that point. Now, the opposite can happen as well, where we have a deletion. One nucleotide is removed. And in this case, again, everything is going to shift over. We have this whole frame shift so that most of those amino acids are going to be wrong after that point. So these insertions or deletions are generally going to be, are often going to lead to complete loss of protein function. Okay, so when are these mutations going to be the most deleterious, the most harmful? Well, first of all, if it's in non-coding DNA, then it's unlikely to have any effect. Okay, really it's where it's DNA that's going to code for a protein. If we see a mutation here, that's where we're going to potentially see some changes to the actual phenotype. Okay, so now let's put some names on some things we've mentioned. Same sense mutations is when there's a base substitution, but one amino acid just codes into the same amino acid. So we're going to have a different base here, but in this case, you can see they both are still going to code for cysteine. And so whether that's an A or a G, this is often going to be the case for like sort of that third spot of the codon. Okay, there's absolutely no change in the phenotype. Okay, that mutation doesn't affect what amino acid sequence is formed at all. Okay, a nonsense mutation is going to be a specific type of mutation where this, instead of coding for a different amino acid, it codes for a stop protein. Okay, now depending on where in the sequence this happens, this could be like a huge issue. Usually the resulting protein doesn't function properly if we stop before we finish the entire polypeptide sequence. Okay, now a missense mutation, okay, is where one amino acid is different and it may have an effect or it may not have much of an effect. So sometimes if the R groups have similar properties between two amino acids, there may not be a huge effect in the resulting proteins. But as we saw with our example earlier of sickle cell anemia, even a single mutation that leads to a single amino acid change can have very severe effects and even lethal effects. Okay, so it turns out a small proportion of these missense mutations can actually improve the functioning of a protein, but these beneficial mutations are massively outnumbered by the deleterious ones because, but they're arguably more significant because they can allow for evolution and adaptation to occur if they're actually helpful to that individual. Okay, it can lead to higher fitness and lead to a higher chance of being passed on to offspring. Okay, so remember we call single mutations single nucleotide polymorphisms. Okay, and this is going to be where we have like one single base difference. And these are going to be found like within a population. These are going to be places where within a population we have different nucleotides at certain places that can lead to then different alleles. Okay, insertions and deletions are less likely than substitutions to have beneficial consequences because they're an entire frame shift. You can see here where if you had one letter different, you could kind of figure out these words, the fat cat sat. But if just we have a deletion and that T is gone, okay, right, every single triplet here is going to not make any sense. And so that is what a frame shift is. All of the amino acids could be different than what they're supposed to be. Now, where does gene mutation come from? What causes it? Well, mutagens. Okay, mutagens are external agents that can lead to mutation, and we have two major types. Radiation. Now, radiation can increase the mutation rate if there's enough energy to cause chemical changes in DNA. So remember that here's our visible light spectrum right here. Remember, towards the blue end, we remember that's the shorter wavelength, which is higher energy. And we remember that beyond visible light then, okay, we have our ultraviolet, which we know can cause skin cancer. It's even shorter wavelengths, which means even higher energy. Okay, and then we get to X-rays, gamma rays, cosmic rays, increasing in energy as the wavelength decreases. And so gamma rays, X-rays, alpha particles from radioactive elements are mutagenic. This is why when you go to the dentist and you need to get X-rays, they put that big heavy smock over you to protect you from overexposure of X-rays. Shortwave ultraviolet radiation in sunlight is also mutagenic, like I mentioned, and can cause mutations in DNA that lead to skin cancer. Okay, some chemical substances cause chemical changes in DNA, and so they're mutagenic as well. Okay, examples are going to be some of the chemicals found in tobacco smoke. Mustard gas is also mutagenic and was used as a chemical weapon in the First World War. Okay, randomness in mutation. Mutations are described as random changes. Okay, it's debatable whether something is truly random, but mutations are certainly unpredictable, and they definitely cannot be directed by living organisms to achieve an intended outcome. Okay, now the consequences of a mutation have no influence on the probability that the mutation will or won't occur. Scientists have not found any natural mechanism for changing a particular base with the intention of making that beneficial change to a trait that's been discovered or is ever likely to be discovered. Okay, we can have mutations occur anywhere in the base sequence of a genome, although some bases do have a higher probability of mutating than others. This is because mutation is a chemical change. Some chemical changes just happen more easily than others. The position of the base within the genome also can affect the chance of mutation. This is because the differences in how coding and non-coding DNA sequences are used and affects the likelihood of mutation. Okay, a mutation is unlikely to be beneficial. Genes are developed by evolution over long periods of time, so a random change will usually be neutral or harmful. All right, so the consequences of mutation vary whether it occurs in a germ cell or a somatic cell. Now, a germ cell is a cell that gives rise to gametes. Okay, so if that germ cell is making either sperm or eggs, and those sperm or eggs happen to be the one that creates the new offspring, then those mutations can be passed on. Somatic cells, though, are body cells. Okay, this is going back to your skin cancer example. If you have a mutation that causes skin cancer, okay, that offspring is not going to be born with skin cancer because it's not a mutation in a germ cell. It's in a somatic cell. So it's eliminated when the individual dies, and therefore it has limited consequences. Okay, mutation is a source of genetic variation. We know that alleles form from those random mutations. Okay, and that's how we have variation in our population, is because all the different alleles all showed up through random mutation. Okay, and we are going to hit this stop sign. So SL students, you are done. HL students, we have just a little bit more. Hang tight with me. Let's finish this up and finish strong. All right, gene knockout. This is a technique to investigate the function of a gene by changing it to make it inoperative. So if we want to figure out what a gene does, because we can sequence DNA, okay, but if we want to figure out what a certain portion of the DNA actually codes for, we can use this gene knockout technique using different model organisms, usually mice. So first, DNA is prepared with the base sequence that allows it to be inserted into the genome of embryonic mouse cells as a target, or as a replacement for a target gene, which is therefore deleted. So that gene is deleted, okay? And we're just going to be putting this other DNA, inserting it as a replacement. Now we're going to take cells where this has been successful and grow them into adult mice. Okay, these adults are only going to have one copy of the target gene that was inserted instead of the two that would naturally have been there because they were deleted in the first place. Then we're going to take males and females that successfully only have one copy, and we're going to breed them. 25% of their offspring can be expected to have no copies of the target gene. They are going to be the knockout mice, and we can then look and see what's missing, right? If we had a gene that, controlled blood sugar levels or some type of brain function, then we can see what's going on in these knockout mice, where if that gene is missing, what can those mice no longer do? And therefore, we can determine what the function of that gene was. Now, we actually have several thousand knockout strains that have been developed by scientists, each lacking one gene, and they've been widely used in research. There are, in fact, now libraries of knockout organisms for all genes in the genome. Okay, use of CRISPR sequences and the enzyme Cas9 in gene editing. So CRISPR-Cas9 is a way to cut and paste, to edit genes, okay? And it's based on a natural system that exists in many species of prokaryotes. So we're gonna see CRISPR regions within the genome, and we're gonna see this enzyme Cas9 used. Now, you don't have to know about how this is used in prokaryotes, but we're gonna run through how it works in this biotech application. Okay, so CRISPR is an acronym for clustered, regularly interspaced, short palindromic repeats. So let's tease out this name. First of all, repeats. The same base sequence occurs several times in the genome, and we see those indicated by R. Here are our repeating sequences. They're short. The number of base pairs in the repeat is only between 23 and 47. Okay, they're clustered. The repeats are grouped in one part of the genome. They are regularly interspersed. They're separated by other base sequences of similar length called spacers, indicated here in these different colors. Each spacer is unique. There can be as few as 2 or over 120 in a particular CRISPR locus. They are derived from viruses that infect prokaryotes, and they allow for viral DNA sequences to be recognized if infection was to occur. Okay, lastly, palindromic. Each repeated sequence has parts which read the same backwards as forward. It's not truly palindromic, but the name CRISPR containing the letter P has stuck, and CRISPR doesn't have quite the same zing. Okay, so let's look at how it goes and how it works. So first, prokaryotes make what's called guide DNA, here indicated in red and maroon. Okay, it's made by transcribing a repeat and an adjacent spacer. The repeat is used for binding to Cas9, okay, which is indicated here in blue. The spacer, which is this maroon part, okay, searches for viral base sequences in DNA. Okay, so Cas9, this blue enzyme, can find those specific DNA base sequences in the genome, okay, using the guide RNA that is bound to it. Again, the guide RNA is this red RNA. The spacer on the guide RNA forms this variable 17 to 20 base sequence at this 5' end of the guide RNA, and it's going to be complementary to the target DNA that is being searched for. The repeat forms the other parts of the gRNA, which are partially double-stranded, indicated here in the lighter red, okay, generating loops and a distinctive molecular shape that promotes binding to the Cas9 enzyme. So, okay, Cas9 is just going to move along the DNA molecule, it uncoils it, and it brings the DNA adjacent to this variable base sequence of the guide RNA in maroon. So this maroon guide RNA is going to search for the complement on the DNA, and it's going to move along the DNA until it finds the complementary sequence. Okay, Cas9, the enzyme, contains 2 endonucleases, and if that target sequence is recognized, the endonucleases cut, okay, the target sequence on both sides of the DNA. It's a double strand break. Okay, now this again evolved in prokaryotes as a way to recognize and destroy foreign DNA within their cells, especially viral DNA. Okay, but this is now allowing scientists to identify specific locations in the DNA, okay, and cut and paste different sequences together. Okay, so we could potentially use it to make important changes to crop plants, to add things like disease resistance. It would allow genetic diseases to be eliminated from human populations. But we need to be aware, okay, that certain potential uses of this CRISPR technology of being able to cut and paste DNA raises ethical issues. And I'm sure you are going to hear more about CRISPR as we move forward, as you guys are the future scientists that are going to be out there using this technology and also needing to make ethical decisions about what's okay to do and what's not. All right, very last thing then for the video. Okay, hypotheses to account for conserved or highly conserved sequences in genes. So it turns out that conserved sequences are identical or nearly identical across a species or group of species. So we see very little change between species for particular DNA sequences. Okay, and then there are some that are highly conserved, which even more, they're identical or similar over long periods of evolution. So they may occur across an even wider range of species. So what type of genes would be conserved or even highly conserved? Okay, there are two hypotheses for why this happens. So first up, functional requirements for the gene products. Many of those conserved species sequences, it's a long video here, are in protein coding elements of the genome. So they're going to be useful. They're going to make proteins that are really useful to those organisms, and so those are going to be conserved between species. Okay, they can also be the genes that are or the parts of the genome that are transcribed to make ribosomal or transfer RNA, or in sequences used to regulate gene expression. These are all elements in the genome that have an obvious function, and so if their function remains unchanged, there's no reason to change the base sequences over long periods of evolutionary time. So we also are going to see some non-coding elements conserved in the genomes, and we don't really understand necessarily what their role is yet. Maybe that's what you're going to figure out. But because they're conserved, that suggests that there is a function. We just don't know what it is yet. Okay, another hypothesis for why sequences are conserved is that some parts of the genome just have slower rates of mutation. We know that mutation rates are not identical throughout the genome. And so those conserved non-coding elements might just happen to be in regions of the regions of the genome where mutation rates are low. All right, thanks so much for sticking with me, and I really appreciate you guys listening.